And so I will transition now to our next speaker, who's Cameron Leitz, who will be presenting his work on interspecific differences in diet among 4 caddisfly species. His advisor is Karen Alos from the School for Environment and Sustainability, and his reader is Hamish Grigg from the University of Maine in Stream Ecology. And so, Cam, we'll turn it over to you.
Let's see if I can get this the screen to share. There we go. Okay, so thanks everyone for being here. So for my thesis, I studied the interspecific differences in diet among 4 caddisfly species. So you might be wondering what a caddisfly is. So caddisflies are aquatic insects that live in the water in their larval stage. And I focused on caddisflies in their larval stage for my study. They often have protective cases to protect themselves from predators. You can kind of see a picture of one on the left here. To go a little bit of the background, detritus, so most plant nutrients and energy enter terrestrial and aquatic food webs as detritus or dead organic matter, dead plants. This is crucial for creating high secondary production in the ecosystem. And so detritivores make nutrients and energy available to secondary consumers by consuming this detritus. Caddisflies are in the northern hemisphere, caddisflies are the biomass dominant detritivores in pond and wetland ecosystems. In this way, they are really good at connecting the brown food web, which is the food web based on detritus, and the green food web, which is the food web based on primate production. And also, caddisflies are also connect the green food web because they are the biomass dominant prey resource as well for predators such as salamanders and fish. So caddisflies live in these ponds and wetlands, and these high altitude ponds and wetlands are limited by nitrogen and phosphorus. And so other organisms rely on caddisflies to mobilize these detritus source nutrients back into the food web. And they do this through by breaking down detritus and through their excretion, which leads to increased nitrogen and phosphorus levels, which in turn leads to increased primary productivity in these ecosystems. So pond and wetlands are also under threat from climate change. And so you kind of see in this figure that caddisflies are shifting their ranges to higher elevations in response to increasing temperatures due to climate change. And then we're also seeing local range shifts in terms of shifting from different pond types, from more temporary ponds that are kind of been drying up more recently to more permanent ponds that last throughout the year. To kind of go into a little bit about the purpose. So our general goal was to examine caddisfly diet in natural conditions and see how it corresponds to their detrital processing and their effect on algal growth in a controlled environment. And so by measuring caddisfly diet in natural conditions, we collected data on their percentages of coarse detritus, fine detritus, algae, and invertebrates in their diet. And then we were able to connect this or link this back to their rate of detrital breakdown and their effect on algal growth that you could see in a controlled environment. And so we collected data from various high altitude Rocky Mountain ponds. And so for our predictions, we said that we thought that caddisfly diet in natural conditions would be maintained when given equivalent food resources in a controlled environment, and this would connect to their detrital processing and effect on algal growth. We also think that, or predicted that the species with the most detritus in their diet in the natural diet would lead to them having the highest rate of detrital breakdown in the controlled environment. And also the species with the most detritus in their diet would have the highest amount of algal growth. Then we also predicted that the species with the most algae in their diet would lead to the lowest algal growth, since caddisflies also graze on algae as part of their diet. So we studied 4 caddisfly species, Anabolia bimaculata, Limnophilus externus, Limnophilus picturatus, and a limnophilid species. That last species is one that we haven't completely identified to the species level yet, due to we haven't reared it to the adult stage successfully. So I conducted my study at the Rocky Mountain Biological Laboratory, which is in Gothic, Colorado. To go a little bit about the methods, so for the diet analysis, so we collected 4 different species of caddisflies from various ponds, and then we collected 10 individual caddisflies of each species and dissected these caddisflies to examine their gut contents. So with the gut contents, we stained them to identify invertebrate material, and then looked at them under a microscope to identify the coarse detritus, fine detritus, algae, and invertebrates. For the experimental part of my study, we conducted inside of a temporary field laboratory, which you can see on the left side of the screen. So inside of this, we had 25 microcosms, which are basically just shoebox-sized containers that are used to simulate natural pond conditions. And inside of these, they're filled with water, detritus, and caddisflies. And at the end of the experiment, we separated the detritus by using a sieve to separate the coarse detritus from the fine detritus. So we determined algal growth by measuring chlorophyll A. As you can see on the left side is a close-up image of a microcosm. And so we were able to measure chlorophyll A by putting ceramic tiles inside of the microcosms. As you can see, there's kind of a mesh enclosure in the middle there, and we put 6 ceramic tiles in the middle of that. And that was to prevent caddisflies from grazing on this. And then we also you can't see in the picture, but we put 6 other tiles on the outside of the enclosure where caddisflies were allowed to graze. This allowed us to see how caddisfly grazing affected primary productivity. Then going to the results, so we used a principal coordinate analysis to determine that there was low intra-specific differences in diet composition. So you can see on the figure on the left, the main point is just to see that each number represents the diet of an individual caddisfly. And generally we see that the individuals of the same species are closely grouped together, even though there are a few outliers in the little circles. But in general, the differences between the same species are lower than the differences among the different species. And then here's just another graph to kind of emphasize the high interspecific differences. And so we can see that there are strong differences between the percent of algae, percent of coarse detritus, and percent of fine detritus among the different caddisfly species. And so we determined significance among the caddisfly species by using a one-way ANOVA test and a Tukey's on a significant difference test. So in this figure, we're looking at on the left side, we see that it's the coarse detritus composition, the caddisfly diet. On the right side is the coarse detritus consumed. And so we found that the natural diet generally corresponded well with the coarse detrital breakdown. So you can see externus, picturatus, and the limnophilid species all kind of are mirrored on both graphs with like high, low, and low. However, we saw that Bimaculata, you can see that it has kind of a medium amount of coarse detritus in its diet, but a really high coarse detrital breakdown rate. So this was interesting, and we thought that this could possibly due to a competitive interaction with externus. And so since it has the high coarse detrital breakdown rate in the microcosms, but it wasn't able to consume a lot of coarse detritus in natural conditions, we think something's kind of preventing it from consuming a lot of coarse detritus in the natural ponds. And we hypothesized that there might be a competitive interaction where externus is outcompeting Vermaculata and forcing it to switch to less food resources that are either less nutritious or abundant. And so this is a graph showing, again, the coarse detritus consumed, but I want to focus on that Vermaculata and externus consumed a really high amount of coarse detritus compared to the other species. And then we also see the box plot on the far right is our control treatment. And so for the control treatment had no caddisflies, but we know that a little bit of detrital breakdown occurs due to, or it's accounted for by microbes. So we have that there. But in general, we see that Vermaculata and externus had really high coarse detrital breakdown rates, which indicate that they're really important for coarse detrital breakdown, or really important for ecosystem function, because coarse detritus is really detritivores are really important for mobilizing these nutrients back into the food web by consuming this coarse detritus. So this graph is looking at the fine detritus generated by the species. So it's kind of similar to the last graph with the coarse detritus. So again, we see that Vermaculata and externus generated really high levels of fine detritus compared to the other species and the control. And so we want to focus on here that also Vermaculata and externus are important for ecosystem function in this aspect, because fine detritus serves as a good food resource for other species of caddisflies and other invertebrates like filter feeders. And so on this slide, we have the coarse detritus consumed on the left and the amount of algae generated per day on the right. And so we know that coarse detrital consumption directly leads to the amount of coarse detritus is directly linked to algae production, because coarse detritus leads to excretion, and the excretion leads to increases in the algal growth. So we expected, based on the coarse detrital breakdown, we expected Vermaculata and externus to both have high rates of algal growth, but we can see on the right that externus has really low algae in their microcosms. This was really interesting. We thought that this was possibly due to Vermaculata having higher concentrations of nitrogen and phosphorus in its excretion, which possibly could have led to this difference. We also saw that, so on the left we have the caddisfly diet composition of algae, and on the right we have the difference in algae generated per day. And so caddisflies incorporate algae into their diet, and we see that Vermaculata and the limnophilid species used significantly more algae in their diet than the other species. And so we expected that, and so the graph on the right shows the difference in algae generated between the ungrazed and the grazed areas. And so we expected there to be a really big difference in that for Vermaculata and the limnophilid species. But we saw no significant differences among the species, which is interesting, which kind of shows that no caddisfly species were consuming algae in the microcosms. This suggests to us that algae becomes a really less important part of caddisfly diet when coarse detritus is really abundant. So overall, we found that the natural diet, caddisfly's natural diet, mostly corresponded with the coarse detrital breakdown rate in microcosms. However, we had an exception with Vermaculata, which was potentially due to the competitive interaction with externus, showing that this is possibly, Vermaculata may be being outcompeted by externus in natural ponds. And we also wanted to emphasize that Vermaculata and externus seem to be the most important species for ecosystem function in these high altitude ponds and wetlands. And we, in terms of coarse detrital breakdown and fine detritus generation. We also saw that pimaculatum was really important for the effect on algae. And so overall, this is really important to consider because caddisflies are shifting their ranges upslope to higher elevation in response to climate change, which is creating new combinations of species. And we don't know, creating unforeseen or creating consequences that we don't know about. And so we can kind of use these species-specific rates to anticipate consequences of these species range shifts and try to predict how different species, or what will happen in these ecosystems if different species are replacing each other. And overall, our work can kind of contribute to creating a model in the future where we can help predict how these changing abundances will affect ecosystems. And there's a few limitations studying that. We had some early emergence and mortality with the caddisflies, and we tried to control for this by adjusting the rates for per day rates, so all of them should have been accounted for. And then we also kept the initial density the same among the caddisflies. And in the future, it'd be nice to have these mass-specific rates by collecting the final biomass data, which we weren't able to get. It would also be interesting to scale up these experiments to a larger scale and to see this. And then I'd like to thank Dr. Karen Alofs for guiding me through this process, as well as a lot of other people in Colorado, especially Dr. Hamish Gregg.
Great,
that's
thank you.
all I have.
Excellent. Well, thank you so much for a great presentation. I think we have time for at least a couple questions. All right, so potentially as other people are thinking about questions and either typing them in chat or unmuting themselves, I'll just ask one as far as you talked at the end about how your work could be thinking about different shifts associated with climate and the caddisfly. I'm also wondering how some of the work could be thought of in terms of other types of disruption. So for example, if there was logging activity or some other habitat disruption that changed sort of the type of detritus that enters into a pond, or the amount, or the types of coarse versus fine, how might that actually play out in some of the interspecies interactions?
Yeah, so one of the things that we kind of thought about a little bit was that one of the big things with the different ponds that the caddisflies live in is that a lot of the ponds are kind of drying up. I talked a little bit about the local range shifts. And so a lot of caddisflies that live in temporary ponds often don't coexist with the caddisflies that live in the permanent ponds. And so we're seeing with climate change, this is causing more drying up, but also some with water withdrawals from increasing development in the area. And so that's also drawing down the water levels, which has potential consequences for forcing some of these species to shift to more permanent ponds where they have to compete with possibly like more efficient caddisflies, but also have to survive a lot of different predators that can exist in these permanent ponds, but not the temporary ponds.
Great. So Shane, I saw that you had unmuted your microphone. I wasn't sure if that meant you had a question. Potentially not. Okay.
I have a question, Cam. So I am by no means an expert on this at all, but how many different samples did you have, and from how many different areas, and how do you think that affected your results?
Yeah, so we collected the caddisflies so we collected all the caddisflies from the same spots, and so the ones from, I think it was 3 different high altitude ponds, and so yeah, we collected them. One problem is kind of transitioning the caddisflies that we took from the ponds into the controlled environment in the temporary field laboratory. We kind of had to carry them down back into the little town where we were in, which might have caused some problems with that. But overall, it was 3 different ponds. I don't think it would affected it. We basically had to choose the ponds based on the availability of the caddisflies and make sure we took them from really abundant areas to not deplete the local populations.
It's cool. It sounds like a lot of hard work.
Indeed it does. So congratulations to Cam. We virtually applaud you. Very nice work and nice culmination of your thesis work in a lovely presentation. So great job. And we